From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 3, recorded on December 16th, 2019. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today here in New York City, Ori Lieberman.
Hi, Vincent.
And we should say now the newly minted PhD.
Congrats.
Thank you. I still have to reformat my thesis. So that's
to reformat. And now you go back to medical school, right?
Yeah, January 6th, first day.
Wow. The day that Jason's here.
Yeah, I know. I might skip out a little bit. We'll see. Yeah.
Also joining us from Nashville, Tennessee, Erin Calipari. Welcome.
Hi.
What's it like in Nashville? Is it warm?
Actually today, so it's kind of rainy, which is gross, but yeah, it was probably like 60 degrees today. It's like perfect. Yeah, I love living here. After New York, the weather makes me very happy.
And joining us from Salt Lake City, Jason Shepard.
Hi, Vincent.
Welcome back, everybody. How's the weather in Salt Lake? I think you got snow out there already, right?
Yeah, we did. We got another nice storm for the mountains over the weekend, so looking good for skiing.
Yeah, I've been in touch with Nels Eldy, host of TWiVO, and talks about skiing. You know, here in New York, when it's winter, it just kind of gets crappy, right? It's slushy.
Yeah, just wet everywhere.
Yeah, you never have nice, fluffy white snow anymore.
Well, gray, fluffy.
Today it's gray and cloudy. I think we're going to get some
tonight, right?
Slush.
Yeah.
Yeah. All right. Today we're going to talk all about an interesting topic Erin's going to tell us about. But before that, if you like what we do on this podcast and all the others, consider supporting us financially. Go to microbe.tv/contribute. Have more on that later. So, Erin, what do we have today?
Yeah, so, we had talked about kind of talking about a little bit about what I work on and kind of going through some things that I thought would be interesting to kind of general science curious people. And so my work focuses on understanding kind of how the dopamine system is involved in information encoding. And so I think one of the interesting things when you're thinking about the dopamine system is you think about, the popular press and what everyone kind of, says dopamine does. And I think you'll see that everyone says, okay, dopamine's the pleasure molecule. It's involved in reward. And then, if you're on Twitter and you follow any scientists that study this, they're like, no, it doesn't. It's way more important than this. And so I figured that of all the things to do, it would be perfect to kind of talk about what dopamine actually does and kind of why people may say that it's the kind of pleasure molecule, but why it's a little bit more complicated than that, and kind of how it's driving our behaviors in a kind of normal and even in maladaptive state.
I think most people know that if you're a boxer and you develop Parkinson's, you get treated with dopamine, right?
Mm-hmm. Right.
Is that correct? Yeah.
Yes. Yeah. And so I think that's the interesting thing, right, is it has a couple roles. One is in motor primarily. And so Parkinson's disease is really focused on neurodegeneration of these dopamine neurons. And so that's in kind of the, thought of as the substantia nigra. And then there's the ventral tegmental area dopamine neurons that are projecting to reward-related brain regions like the nucleus accumbens that I study. And it's more involved in encoding information about kind of associations between predictive cues in the environment and their outcomes. And so, the reason we're interested in this is because, one of the most fundamental aspects of survival is the ability to associate positive and negative outcomes with stimuli that predict these. And so, a few examples are things like you put your hand on a hot stove, you learn very, very, very quickly not to do that again. And so, what's happening in your brain that's helping you not do that again, but also is helping you do things that result in positive outcomes. Like you go to an ice cream truck and you get a reward. How do you know to do that again? And so, these are kind of at the core of every decision we make. And we use this kind of information, and that the paper we'll go over today kind of gets at this topic. We balance the probability of good or bad outcomes with one another to kind of make decisions about what we're going to do. And so, for example, you need to know how to eat because eating is critical for survival, but you also need to know when to stop eating and how to not make yourself sick from doing that. And so understanding the kind of positive and negative balance is such a critical component of nearly like every single aspect of what we do. And then when you take a step back and you say, okay, well, that's like on a basic science level, but that is exactly what's dysregulated in almost every psychiatric disease. And so when you're thinking about things like anxiety, stimuli in the environment that signal something kind of negative or overgeneralized. And you think about depression, you have a reduction in like the attribution of reward to kind of stimuli in your environment. For drug addiction, you have a dysregulation of this balance where one, the drug-associated stimuli are overvalued relative to everything else, but also so are the negative outcomes. And so people take drug in the face of negative consequences, even though they know that this may result in something negative, they still take drug. And so this kind of balance between, positive and negative stimuli and how it's guiding our actions is a really critical component of basically every kind of, fundamental behavior. So that's why we're excited about it, because I think it's kind of at the core of everything we do.
I'm actually curious about where this sort of simplistic view of dopamine equals reward in the general public came from. Is that from those old studies where rats were just pressing a lever for a reward, and if you substitute dopamine, I can't remember if that's
yeah. So there's so much. I think a lot of it is probably popular press and scientists, all of us are kind of guilty of this, is like overly simplifying our data into some really nice conclusion with a bow on it. Yeah. So animals will press a lever for basically indefinitely for stimulation of these dopamine neurons. If you block dopamine receptors while they're doing this, they won't do that anymore. The kind of stuff we'll go over today, one of the big theories of what dopamine does is reward error kind of encoding. And so I think all of these things have really kind of come together to say, okay, well, what dopamine does is it encodes reward. But I think there's a lot of evidence in the literature that dopamine isn't really just a reward encoder, that it actually responds to things that are important in the environment. So it's almost kind of like there's two different fields that haven't communicated with each other well. There's tons of data also showing that aversive stimuli increase dopamine, stressful stimuli increase dopamine. And so when you think of like, depression, there's dysregulation of dopamine as well. And so, even if you look at, I think part of the problem was with physiological techniques that rely on electrical recordings. A lot of our aversive stimuli in animal models are based on, shocking the animals, and you can't kind of pair those together. But when you look back at the microdialysis data, which is kind of just more long-scale dopamine detection, you do see increases to aversive stimuli. And so dopamine responds to essentially everything that's salient in the environment to kind of help animals navigate an environment. And if you get rid of dopamine, they can't do this for any stimuli. And so I think it's kind of an important thing to understand that dopamine's not just, more dopamine good, less dopamine bad. It's more of a complex regulation where dopamine's responding to appropriate things at appropriate times. And then that's helping animals decide kind of what to do in conflicting situations, in situations where they have to decide whether they inhibit a behavioral response or they emit an action. And so I think that it's more of a complex story, that with all these kind of new optical imaging tools, we're starting to kind of understand more of a holistic view of how this is working. Say
a dopamine neuron. Does that mean they produce it, respond to it, or both?
Both. So my lab actually studies both of these things. So the dopamine neurons are really defined by the fact that they release dopamine as a neurotransmitter.
All right.
And so these neurons originate mostly in the ventral midbrain, and so the substantia nigra and ventral tegmental area, and they project throughout the entire brain. And so they're essentially innervating almost everything. And so, one of the places that we've studied is the nucleus accumbens. And so, in the nucleus accumbens, where dopamine neurons are projecting, the dopamine terminals from these midbrain neurons release dopamine. And this can act on a number of kind of different receptor subtypes in the area that are either located directly on the terminals themselves. So these function to kind of as a feedback mechanism. So dopamine's released and it acts on these D2 autoreceptors. And what it can do is shut off future release. But it can also act on postsynaptic dopamine receptors that are located on nearly every single cell type in the area. And it acts to kind of gate the kinds of information that's being encoded through different kind of glutamatergic inputs into the region. And then to overcomplicate things, dopamine is actually also released from cell bodies of dopamine neurons in the midbrain. And then that dopamine release happens actually through different synaptic mechanisms than at the terminals. And then that dopamine can actually feed back and act on that dopamine cell itself to kind of suppress firing. So these dopamine cells are kind of interesting cells that have multiple different modes of regulation, and plasticity in any of those processes can lead to differences in the way the animals are kind of, encoding information in an environment. And so there's a lot of cool ways to look at all of these things and how they're integrated. I think the biggest probably field in this regard has been in the drug addiction field, which is one of the place that I probably am most comfortable, because almost every drug of abuse, and I think this is where this kind of reward hypothesis comes from, is almost every drug of abuse acts directly on this dopamine system to increase dopamine. And without that increase, these drugs you don't see animals transitioning to these kind of dependence like behavioral states, and they actually won't consume these drugs. And so this dopaminergic component, the ability to increase dopamine, is the kind of critical first step in why addictive drugs are so addictive. And so a lot of the work has looked at how those drugs act on the system, how they modulate ongoing activity, what their pharmacological actions are, and they all do it kind of differently. And so, it's a kind of interesting system because of its role in kind of all these complex behaviors, and then also the drugs that we have that act on it.
I guess it's amazing that drugs with so many different targets can all work through dopamine, right? Mm-hmm.
Well, and I think that's always been the kind of interesting thing in the substance use disorder field, is that, you have things like stimulants that act directly on the ability of dopamine cells to clear dopamine. And so those are kind of really direct actions. But then you have opioids, alcohol, nicotine, all of these act through either local interneuron circuitry at the level of the terminal or the cell body that can modulate how much dopamine is being released at a given time. And so I can't think of any drugs of abuse off the top of my head that don't have some effect on how the dopamine system is responding to either environmental stimuli or just like result in enormous amounts of dopamine release themselves.
So can I ask a quick question?
Yeah, of course.
So these cells are sending their axon terminals quite far away from the cell body. So do you want to maybe talk about briefly like local regulation? So does an action potential fired in the cell body always lead to the dopamine release in the terminal?
I feel like you might know the answer to that one too.
That was a leading question.
Yeah. So Ori and I are actually in the kind of same field. He's more on the mechanistic side than I am, and I'm on the circuit side. So I think one of the fascinating things for me and in my lab about the dopamine neurons is this ability of these local circuits in projection populations or in these projection regions to directly modulate these dopamine terminals. And so, when we think of action potentials in a glutamatergic cell type, for example, you learn like the action potential, it occurs, it propagates down the axon, there's calcium influx from voltage gated calcium channels, and then the neurotransmitter is released, and this is how this works. The dopamine system is a little bit more fun than that. I'd say complicated, but I think it's more exciting.
Definitely more fun.
And so,
more rewarding.
Yeah, I love it.
Waiting for that.
Nailed it on that one. Yeah.
So the interesting thing there is there's a number of ways that you can evoke dopamine release totally independent of VTA cell body firing. And so there's cholinergic interneurons in the nucleus accumbens directly that if you stimulate them, you can actually cause dopamine release from the terminals that is totally dissociated from the VTA cell body firing. And then there's a number of ways that you could also modulate ongoing inputs to change the probability of whether there'll be release of neurotransmitter or not. So things like GABA receptors on these terminals, things like kappa opioid receptors. So dynorphin is released from postsynaptic neurons and feeds back and can act on these terminals, as well as differences in ligand-gated ion channels like nicotinic receptors. And also there's a number of different calcium channels that can be regulated independently of one another that can alter the way that these neurotransmitters are released. And so, one of the interesting things in the dopamine field is a lot of the work on what dopamine is encoding comes from cell body activity. And so people have done electrophysiological recordings in the cell bodies, and they said, okay, well, this is what the firing pattern looks like of these neurons. It goes up to this and down to this. Therefore, this is what the dopamine release is doing at those terminals. And then when we've gotten tools to actually record from those dopamine terminals, in some situations, that's exactly what's happening. But in many other situations, it's a little bit more complicated where neuropeptide systems are coming in and really shaping how this dopamine signal is transmitted. And there's a lot of data showing that, these calcium influxes into these terminals in these dopamine, with these dopamine vesicles don't always mean that vesicle will fuse. So just because you have calcium flux doesn't mean that those dopamine vesicles are definitely being released. And so there's also complex regulatory mechanisms at the level of the kind of vesicle that dictate which vesicles will be released when, that is even kind of more complicated in these other processes. So the system's really interesting because it gives you a lot of ways where you have some event coming in, but it can be modulated by things like internal state, stress, other kinds of things that we know affect these neuropeptide signaling or these other circuits-based mechanisms that can kind of shape how that dopamine signal coming in is ultimately transmitted downstream, which is actually what gets me so excited about the system because it's really complex and there's a lot of cool ways to kind of study all of these different circuits.
So what we're saying is that scientists who get excited about their work, that's dopamine based, right?
Oh, definitely.
Sure, yeah.
There was a paper that came out not that long ago showing, I forget what it was. It was like an fMRI study showing kind of reward-based neural signatures when papers were accepted in scientists. I'll have to find it and I'll send it to you guys, but I forget where exactly it was published, but I thought it was hilarious because I'm like, yep, that's exactly what I would expect from us.
So people like yourself who are studying addiction and drugs, is one goal to get enough of an understanding of what's going on so you could treat it?
Yeah, so I think the one issue, yes, so that's the ultimate goal. And I think that most of us are trying to say, okay, well, let's understand this process at a really basic level to figure out what the molecular underpinnings of this drug-induced plasticity is and how we could reverse it. I think the thing that a lot of us have run up against is that drugs of abuse, what they do is they increase dopamine, we can say that, but really what they're doing is they're modulating normal learning mechanisms. So let's say, in a normal environment, you learn about some cue that predicts a positive outcome. So now you've associated that environment with that outcome. This is like the normal way you'd be able to, avoid or seek out rewarding stimuli. So that relies on dopamine. So dopamine is released when there's unexpected salient events in the environment. A lot of times people look at this in the context of reward. And what that allows us to do is associate whatever those predictive events leading up to that were. So you can associate a room or an ice cream truck or something with a positive outcome. And so these are really important processes. What drugs do is they don't just increase dopamine, they act on the way this dopamine system functions. So let's say you take drug in a specific room. Okay, well, what dopamine is increased pharmacologically by that drug. So what you do is you associate that room with that positive experience of taking the drug. So this happens every single time you take the drug, over and over and over again. And what you've done now is you've enhanced a normal learning process. So you get plasticity in the system, but the problem is, how do you prevent or reverse plasticity in that system without affecting other normal behavioral outputs? And this has really been one of the big limitations in the addiction field is actually twofold. One, we have some drugs that can reduce, drug taking and relapse, but almost all of them have abuse potential on their own. And then 2, if we block the dopamine system, there are so many off-target negative effects that the compliance with drug taking, with the medication taking is essentially very low. And so how do you kind of go and understand a system that's really critical in nearly every aspect of behavior and kind of fix that problem? And I think that's kind of that's the magnitude of the challenge we're up against, is figuring out different types of plasticity that are selective to the drug experience that don't overlap with other experiences. And it's incredibly difficult with the kind of resolution we have to figure out how that works.
So I guess methadone is an example of that, right?
Yep.
Originally made to deal with heroin addiction.
Yep.
But it can be addicted itself.
Right.
Yes. Yeah. And so I think methadone is an interesting one because it's actually really effective.
Mm-hmm.
And so, you take methadone, it's a partial agonist at mu receptors instead of a full agonist. So what it does is if you take heroin when methadone's on board, you block the effects of heroin because it's already occupying the receptors.
Mm-hmm.
And if you basically have methadone on board, it prevents withdrawal effects from the drug. So the people don't have the adverse effects of the withdrawal. And if they were to take drug in a situation, they are less likely to overdose. And so the reason methadone was approved is because it reduced fatalities. And it was really effective at doing this. The problem with stimulants is that the things that reduce drug taking don't reduce fatalities. And so, you still have overdoses, but the overdoses from stimulants, even though they do result in death, sometimes it's a lot less than opioids. And so, amphetamine is actually a great example. Low-dose amphetamine can actually reduce drug taking. It can increase, clean urines, but it has abuse potential and it doesn't reduce fatalities. And so this kind of replacement therapy angle hasn't worked in the stimulant field. And then we really don't have a lot of other drugs that have effects on these kinds of that are effective. Naloxone seems to work pretty well for some people for alcohol abuse, also for opioid overdoses. And then there's Chantix works for some people for smoking. That's varenicline, but not all of them. And so I think right now in the addiction field, it's really difficult. We just don't have a lot of medications. And also most of the stuff we do that would target the dopamine system either is bad off-target side effects or addiction potential. And so I think that's the big issue that as a field we're up against, and it's hard to solve.
Is there a sense that it's unsolvable?
I don't think that's the case. I think that we're starting to get better genetic ways to do genetic screening. I think with all the big data approaches coming out, I think we're going to have an easier time identifying new targets that could be efficacious. When you're thinking of bioinformatics and the way to take patient population data and overlay different kinds of information from sequencing data to patient outcomes to vulnerability data, then I think you'll start to identify different risk factors and things that could be targets that were really hard to do before if you didn't already kind of know what you were looking for. So I'm kind of hopeful that these kind of approaches where we say, okay, we have ability to record thousands of neurons in a single organism, in a behavioral task, and we can do whole genome sequencing on the DNA or RNA or chromatin conformation or DNA methylation across the entire genome in an individual organism. And we start to get all these data sets and say, okay, well, what are the signatures that really predict the different kinds of drug taking? Then I think we'll get closer to saying, okay, well, what targets should we be targeting that may not necessarily directly interfere with normal learning processes? I think that's my kind of hope. I think we're getting there. I think it's just psychiatric diseases are a slower process.
That's something I was always fascinated by, is that even in rats and mice that are, pretty homogeneous genetically, you have sets of animals that just don't get addicted, right? And then you can breed them. So you can breed the mice or rats that are not addictive, and then you slowly get a homogeneous population that seemingly are not addictive. So I feel like that approach of seeing where the genetics is pretty fascinating.
Yeah, I totally agree with that. I think, and there's a lot of evidence in the literature for different kinds of either epigenetic or genetic factors that make people either particularly resilient or susceptible to addiction. And I think that's something the addiction field is really overwhelmingly focused on what makes individuals susceptible. But I think another avenue going forward is figuring out what makes particular individuals resilient. So why we've always focused on why did this, does this, I forget what the statistic is, like 10% or something of people who use drug ultimately become addicted. Well, what happens with the other 90? Right. You know what I mean? What is it about those individuals that they can take drug all day and not develop dependence or a problem? Can we actually use our understanding from that side to maybe develop targets that are able to help the other individuals? The genetically. Oh, sorry.
I was going to say,
yeah.
No, I was going to say another important point I think for listeners, right, is that you see a lot of this in the discussion about addiction, that there's clearly prior history, there's traumatic events that sort of predispose people, but in the end, once you are addicted, it's a disease, and I think that's that aspect of it is lost. And so people are like, oh well, that person is addictive because they're lazy or they just don't want to make an effort, but it's actual physical changes in your brain that alter this. And so that's real clear. I mean, every time I talk to someone who's sort of interested in neuroscience, that's one of the first things I tell them is that, these brain diseases are actually diseases, just like, a disease in the muscle.
And I think this is a big thing, especially, my family's in Kentucky, I'm in Tennessee. I think that if you look at kind of the cultural view on what substance use disorder is, places where the view is that addiction isn't a disease have way worse outcomes than other places. And so I think one of the really important things is that people don't seek treatment when they don't understand it's a disorder. Right. And it's such a hard thing to break because it's something that's so integral to kind of our daily lives and where you're taking drug and who's taking drug and where drug's available. And so it's causing changes in your brain that make it almost impossible for you to not continue taking drug. And how do you kind of inform people that this is such a critical part of the disease? And I think that's been, I think science communication and things like that are really important, especially for this. I think the example I always give is if someone smokes cigarettes and gets cancer, no one's like, oh, you did this to yourself. People are like, that's terrible. And so I find it very strange with other drugs that people take drugs and some of them become addicted and people are like, that's your fault. Because It's a very similar process, right? It's the same thing. It's like you couldn't have possibly known that would happen, and it doesn't happen to everybody. And there's a lot of anecdotal evidence that people, smoke for years and don't get cancer, or take drugs forever and don't become addicted. And so I think I think that complexity makes it harder for people to understand how dangerous it really is.
And the other thing I'd add is, so it's not all completely depressing, I guess, is that there's a lot of behavioral approaches that have promise, right? As opposed to kind of molecular or classic drug approaches in treating or increasing drug-free periods, right?
Yes. I think this is the biggest thing, is that they're behavioral interventions that work really well, but then the issue is that you have to have healthcare and have access to these. Yeah. And so I think this has been one of the big issues, is that until people see this as a disease, then it's hard to get policies to have them basically enact the access to this. And so I think everybody kind of wants this, like, oh, what's a pill that's going to cure me? And I really don't know if it's going to be that, right? That we just have one pill and you're cured forever and you kind of go on your merry way. I think it's going to be more complex. And I think it's exactly what you were saying, is like behavioral interventions where people kind of have to understand like what their triggers are, why this develops, how they can kind of manage these things. And I think understanding how the brain works, how plasticity in the brain is important for how you learn information, is actually a really critical part to understanding why you should avoid this environment. Why, if you have alcohol use disorder, you probably shouldn't be in bars. You know what I mean? These are such really important points, of like educating people on like what's going on and why helps people to understand why they should be kind of doing things in that specific way.
Yeah, for sure.
When we talk about addiction, is it always to a chemical, or can you have behavioral addiction as well?
That's a great question that I think that there's multiple sides to this argument or this debate. I think most people are coming around to the idea that people can have addiction and I fundamentally believe this, to other, natural substances, things like gambling, sex addiction, food addiction. I think these are really, really important things because they show the same kinds of compulsive behaviors. They show the same kinds of kind of inability to stop their actions once they've initiated. So yeah, I mean, I think you definitely can. The question is whether the basis is identical. I think the big thing too for a lot of these disorders, even for other psychiatric disease states, we've started to see that a lot of these disorders are on a spectrum, and it's not just one size fits all. And so I think those definitely fit under the umbrella of not substance use disorder, but addictions. But if they need to be treated differently, although I think one of the really interesting things in kind of our work is we look at sex differences is actually a big angle that we kind of focus on. And one of the things that you'll see if you start to look through the literature is that in males and females, kind of why they took drugs in the first place can be very different. And so, you see more often than in women taking drugs to avoid anxiety, to avoid kind of depression and such stressful situations, where in men, you'll see a larger proportion kind of impulsively taking drug in social situations. So when you think about this and you think about comorbidities, there are really big differences in certain types of substance use disorder in different individuals, right? If somebody's taking drug because they're trying to avoid some traumatic stress from their, childhood, you would probably treat that very differently than someone who, was binge drinking in college without that underlying pathology and just kept drinking into their adulthood and now has another problem. There is very serious overlap between those, but there's also very different kind of core factors driving this, and understanding that is a really critical component of kind of these disorders as well. And so I think it becomes complicated because each individual has a different experience, and each kind of experience contributes differentially to kind of why they're doing these behaviors. And then when you think about stress interactions, stress can trigger relapse. And so these kind of interactions are really critical to understand. And so these disorders, even though they fall under the same umbrella, can all be very different. And I think that's kind of part of the difficulty in just doing like a one drug solves the problem approach, because it's not all exactly the same.
I'm interested in the methods to study this. And I guess your paper kind of gets into that a bit, right?
Yes. Yeah. So I think that there's a lot of different methods to study this. I think one of when you're talking about substance use disorder, we can start there because I think there's a lot of really good, it's behavioral models. So, when you're thinking about taking drug, making decisions to take drug, what factors influence this, one of the things that's really nice about this field is that the behaviors that drive that are conserved across species very well. I think with some other psychiatric diseases, it's very hard to develop models in rodents that kind of recapitulate the complexity of the disorder in humans. That's also true in some cases for substance use disorder, but at its core, it's the motivation to take drug. It's changing how that happens. It's changing how it interacts with alternatives. Operant conditioning, where animals, essentially what we do is we put them in operant boxes. These boxes have levers or nose pokes, things that the animals can interact with, and they're hooked to a computer interface so that the animals can essentially give themselves drugs or rewards of other kinds. And so there's been a lot of work since, the 20s looking at kind of how these kind of behaviors manifest and what kind of factors in the environment control these behaviors. And so, it started early on with like Skinner and pigeons. And so pigeons are really good at pecking keys to get food. And then, it's kind of happens now, people look at it across mice, rats, basically almost every single organism I've seen. There was an operant task in honeybees recently that I saw that was published in a good journal, looking at how honeybees could learn to kind of interact with their environment to get rewards. And so there's really great behavioral models for understanding kind of how these behaviors manifest. And then once we can do that, then you can go in and say, okay, well, now that we have a behavior we understand and we can manipulate it by changing the different kind of parameters in this chamber, how can we record from neurons in the brain while the animals are doing this to understand what those neurons are actually encoding? And so I think that the ability to kind of interface these two kinds of technologies, and so one is operant conditioning with something like electrophysiology, where you can record the firing of these different neurons, has been a really powerful tool for extending our understanding of what cells in the brain are really doing to guide a behavior. And so I think that's the kind of exciting thing here, is interfacing recording techniques where you can record from cells in the brain, techniques where you can manipulate those cells. So now we have tools like optogenetics, where you can use light and these light activated ion channels to cause cells to fire, and complex behaviors. We can kind of close the loop and say, if we stimulate these cells, what do they do to behavior? If we record during behavior, how do these cells behave? And then we can start to map those onto kind of bigger models of how the brain behaves in specific environments to guide these behaviors.
So I guess... Go ahead.
Oh, sorry. No, I was going to just mention this as an aside that my first research project ever as an undergrad was on pigeons and putting them in this operant chamber, and they had hippocampal lesions. So this was actually just looking at straight learning and memory. But the funniest thing was that the way you transport the pigeons from their cages to the operant boxes, you literally put them in a jug, like head first in a jug. As soon as they're in the jug, they don't struggle. And I remember like seeing this for the first time. It was just like the most bizarre thing I've ever seen. But anyway,
that's actually I've never actually seen pigeons in action, but all of the background for my graduate training, there was a bunch of classes on behavioral pharmacology, and they're all of the early studies are pigeon self-administration. Yeah. They're actually really good at it.
They are
probably better than mice, which are not as good.
So I guess in some cases you would put electrodes into the brain, right?
Yeah.
And you mentioned functional MRI earlier. Is that another way to look at what's going on, or is that less detailed information there?
I think that's a little outside of my area, but you can definitely do that in tasks in the scanner. I think it becomes a little more challenging, but there's a few people here at Vanderbilt that do that during different kinds of, they show visual cues and they look at what brain regions light up. I think you lose a little temporal information relative to these kind of physiology techniques that are really fast.
And spatial.
Spatial. No, exactly. But what you lose there, usually if you're doing fMRI, it's in humans. And so, you kind of have the human data, then you can do stuff in primates and rodents, and you kind of tie these all together to say, okay, well, in humans, these are the brain regions that are active when people see, drug-associated cues or reward-associated cues. And then you can go into these other kind of model organisms and say, okay, well, in that brain region, if we show these animals these cues, how is this actually functioning? And there's an enormous amount of overlap between what you see on these slower timescales and these imaging techniques in humans with the kinds of things that we look at in these kind of primates and non-human primates and then rodents as well. And so I think it's kind of nice to tie these all together because we have these tools across fields that allow us to kind of see how that goes in a more comprehensive way.
So you picked this paper,
I did.
Discrete coding of reward probability and uncertainty by dopamine neurons. Tell us about this. Why did you pick this particular one from 2003?
So the reason I picked this, so the original paper showing how dopamine neurons respond to kind of reward predictive cues came out in 1997 by Schultz as well. And it's actually a really interesting paper, but most of it is actually very computational. And so I thought that might not be as good for a discussion for general audiences. And I said, okay, well, let's look through these papers and kind of see something that's a little bit more simple, but kind of highlights exactly how these cells respond in changing environments in a way that has been really critical for kind of informing how the field has gone forward and in understanding these dopamine cells. And so this paper, it's a fairly simple paper. Essentially what they did in the paper, so this is in monkeys. And so I think one of the powerful things about a lot of the work from this group is it's in primates. And this is kind of a really powerful system because there's a lot more homology between us and non-human primates than there is between rodents. But a lot of this work since then has been kind of replicated in rodents as well. And so we kind of see these exact same patterns across the dopamine system, irrespective of species. So essentially what they did is I talked a little bit about earlier about how we associate cues in an environment with an outcome. So you have in these experimental models, you make this incredibly simple. You say, okay, what we're going to do is we're going to give a visual cue, and when that cue is presented, a juice reward will be delivered. And so monkeys love juice just like we do. And so this is a really powerful way. This is called Pavlovian conditioning. And so the question is just kind of what happens when a cue comes on and reward is delivered. And so what happens here is they started kind of from there, that the reward, in that case, the cue is always predictive. But in a normal kind of environmental, situation, cues and rewards aren't always one-to-one. There's some probability that this will happen, right? So there's cues that always predict some outcome. There's different cues that predict an outcome will happen some percentage of the time. And then there's cues that predict that an outcome won't happen. And so there's all kinds of different complex cues that we're integrating. And this kind of probability-based computations that we're doing in the brain are really critical for informing the likelihood of something happening and what we should do in those cases. So, the first thing they did in the paper, and it's in the first figure, is what they did is they increase reward probability. So what happens is, again, this cue comes on and a juice reward is delivered.
What is a cue, by the way? What kind?
It's just a visual cue. A lot of times, so in rodents, we'll use auditory cues, but it's just a stimulus in the environment. So it's a conditioned stimulus. So essentially what happens early on, this stimulus doesn't necessarily have any value itself. So it could be like a square on a screen. In mice, we use some visual cues, but also auditory cues. The cues are essentially, they start out as completely neutral. They don't mean anything. And over time, as you associate them with something positive, the cue itself will start to acquire value, the value of the associated stimulus. And so what they did is they did that simple conditioning experiment, but what they did is they increased the probability that that cue would predict the reward. And so, on the lower end, they had a probability of 25%. So 25% of the time when that cue is presented, a juice reward is delivered, all the way up to 100%. And so what's really interesting and maybe not surprising is that when you give that cue, the monkeys will actually start to lick before the juice is delivered because they anticipate that it's going to happen. And so this anticipatory licking actually increases as the probability goes up. So if something is 1-to-1, the probability of that animal licking is very high. But if something is very low, the animal will still lick in anticipation sometimes, but it's much lower. And so it's kind of the behavioral measure to show that the animals understand the kind of probability-based, cue information here.
So if you have a cue that only gives a reward a quarter of the time, they're not going to always lick.
Exactly.
Or they lick for shorter amounts of time.
Yeah, exactly. Or they lick for less amounts of time. So they may not always lick. They lick for a shorter amount of time. In rodent studies, there will be slower latencies to respond. So the cue comes on, the animals will respond, but it takes them much longer. So these cues are less potent drivers of behavior, which is maybe not that surprising. And that's how we can kind of guide our behavior in a context. If something is really, really strongly predictive, then you know that you need to actually act. But if something's kind of weakly predictive, then you make a decision, okay, is it worth it to act? Is it not? And so this kind of probabilistic decision making is a really critical part of how we kind of guide behavior when there's conflicting information in the environment about what to do and where to allocate our effort. Yeah. So they started there, and essentially what they did throughout the study is they just said, okay, well, in these different cases, let's just record dopamine neurons and see what they do to these different stimuli, the cue and the outcome. And so, the early work by Schultz in these primates, what they show, which is really interesting, is that early on, if there's an unexpected reward, so a reward comes, no prediction at all, what will happen is that you'll get a dopamine spike to the unexpected reward. But over time, as you've paired a cue with that reward, the presentation of that reward, and we're assuming here that it's 100% prediction, what will happen is the dopamine response will actually transition to the cue itself. And then when the reward comes, nothing happens because it was predicted. And so essentially what happens is it's a way that this dopamine system can actually guide what's expected and unexpected in an environment. And so in the early studies, what they did is they had this cue that predicted the reward. When it was delivered, no change in the firing rate of VTA dopamine neurons. But if the reward was predicted and they omitted the reward, what would happen is you would still get that initial dopamine response to the cue because the animal predicted the reward would come. And when the reward didn't come, then there was a depression of the dopamine cell firing. And so what the system does is it acts to kind of encode these kind of predictions, but then update them when something has changed. And so if the reward was better than expected, the dopamine neuron firing to the reward goes up. And if it's worse than expected, the firing to the reward goes down. And so what they were doing here was kind of playing off those original studies and saying, well, does that kind of hold true in the case where the probabilities are changing? And so in the second figure, it's really interesting. What they show is that as you increase the probability that the reward will be delivered when the cue is presented, that the cue responses, the firing rate of the dopamine neurons to the cue actually go up to the cue as the probability goes up and down to the reward because there's less of an expectation deviation. And so in the cases where the probability is lowest, the response to the cue is the lowest and the response to the reward is the highest because there's kind of more surprise. The animal didn't expect it to be there or there was less of an expectation. And what that does is it leads to an increased response when the reward is delivered. So there's more of this kind of surprising situation. They kind of went through and characterized that, just showing this kind of both with traces, but also empirically that essentially your response to your reward goes down as the probability increases, and the response to the cue goes up as the probability decreases. So the dopamine response to these two stimuli, kind of the balance between those, is a way to encode information about like how probable it is that a reward is coming when there's something in the environment that does that.
So I'll ask a quick methods question here.
Yeah.
So these are electrical recordings of putative dopamine neurons.
Yes.
So, and in this case, how do they decide that they're dopamine neurons? And also, I think that in this paper, they combine recordings from different areas of dopamine neurons. So how do you think about that?
Yeah, so that's actually a great question. So I'm assuming, I don't do primate physiology, so I don't know exactly how they do it, but I have done it in rodents. And so dopamine neurons have a really specific waveform and kind of spike timing that allows you to dissociate them from other cells in the area. So I'm fairly certain
that's what
that's what they did.
Yeah.
And then, so for this paper, they kind of grouped different populations of neurons. And I think there's a lot of data coming out now. This is kind of the early work that different dopamine neuronal populations kind of respond to different kinds of things in the environment. And so, there's some work from other groups showing that these kind of prediction-based neuron firing patterns only occur in like 30% of dopamine neurons. But there's still quite a large percentage of them relative to the information that's encoded in the rest of them. So this pattern has been seen in the ventral tegmental area. It's also been seen in the substantia nigra. And this pattern has also been seen in in nucleus accumbens recordings of dopamine release. And so there's a lot of work where people have used techniques like fast scan cyclic voltammetry in vivo to kind of redo these experiments in rodents to understand if the dopamine being released at terminals in projection populations actually tracks this pattern. What I'll tell you is that in reward-based contexts, it definitely does. In context where the information is negative or there's aversive stimuli, there seems to be some divergent kind of dopamine release patterns suggesting maybe local circuit regulation that happens a little bit differentially in those cases. And so, this was like the early work saying, okay, this is what happens in a large population of these neurons. But then now I think people have gone in to really refine that with different kinds of genetic recording techniques where we can label specific projection populations of VTA dopamine neurons or get cellular resolution so we can see the neurons themselves with techniques like in vivo calcium imaging. People have done a lot of work now with optogenetics to stimulate these different kinds of projection populations in different regions to look at how they affect reward seeking versus orienting responses versus associative learning. And so, it's these papers, as always, and a lot of these bigger journals are all these kind of first step simplistic understanding, and then we go through and we refine it further. So I think there's a lot of information coming out now, especially with new tools, to kind of really understand what these cells are doing relative to kind of other cellular populations in the area.
Cool.
Yeah. And so, oh, sorry, was somebody saying something?
No, I just said cool. Yeah.
No. This is the thing, though. But I think this kind of gets back to the thing about dopamine is encoding reward. This kind of probabilistic learning also happens for aversive stimuli as well, and dopamine seems to be playing just as an important role in that kind of learning too. And so thinking of dopamine in the context of just reward encoding, I think is really overly simplistic. But here's the thing, it does encode important information in the environment. Stimulating dopamine does support behaviors that we associate with reward. And so I think that there is some kind of validity in understanding dopamine as something that's really critical in the kind of the ability of rewards to drive behavior, but it's a little bit more complicated because it's more of an encoding of kind of coincidences between environmental stimuli and rewards rather than just the reward itself, which is why I think it's such an important thing to kind of to talk about.
And then, of course, the one of the interesting questions is, at the molecular level, what is dopamine actually doing to change the properties of the brain? I guess, Ori, that's some of what you're doing.
Yeah, vaguely. Dopamine receptors are GPCRs, and so they have a lot of different signaling cascades that are activated, and this can change synaptic strength, excitability of the cell, of the postsynaptic cell, and obviously change kind of circuit function after plasticity and stuff like that. But it's very complicated, I think. Yeah.
Well, yeah. I mean, Erin came from one of the best labs in the world for looking at this, in Eric Nestler's lab. So, it's that lab's done an amazing job of sort of parsing out what genes are induced by these various drugs.
Yeah, well, and I think that's the really interesting thing too. When you think of kind of dopamine as an encoder, I think that the dopamine field sometimes also is, and again, I'm one of them, has a little bit of an overly simplistic view of what dopamine is, just because it's a neuromodulatory transmitter. It doesn't bind to ion channels and induce firing directly in postsynaptic neurons. It's binding to G protein coupled receptors that have different spatial distributions based on the G proteins that are signaling through. So dopamine has this kind of really potent role in the ability to induce different transcriptional patterns in different cellular populations in the same spatial location. And so I think that there's a really important information there on how dopamine is causing changes in different cell populations at a given time and in their ability to respond to other kind of glutamatergic or GABAergic inputs from that area. So I think that when you look at these kind of dopamine, just going up and down is interesting, and we look at that too. But what it does postsynaptically isn't just up or down, right? It's up or down in a specific cell type expressing a specific type of dopamine receptor if there is also an input from another population of cells. And so it kind of provides this really cool filtering mechanism where it can filter for specific kinds of information coming in at specific times rather than just encoding the information itself. And so I think that's why it's such an interesting neurotransmitter system and why its dysregulation in disease has such important implications for the ability of other systems in the brain to function.
And that's why it's interesting that the genes, many of the genes that are modulated by dopamine, of course, as you know, are critical for just normal learning and memory. And so there's this other idea that addiction is just some form of memory or learning that's abnormal. So if this is where our fields, are intertwined, where it's a lot of these genes that are induced just by simple learning are modulated by drugs of addiction.
Right. Well, yeah, and I think this is what makes addiction so hard to treat, right? Is that you have these like simple these things that you learn and it changes the transcriptional activity in a cell, but addiction kind of does the same thing just to a bigger magnitude. I think I'm excited about epigenetics as a kind of understanding addiction, because I think that when you have these really strong stimuli like drugs that pharmacologically induce a signal that's really above and beyond what would normally happen in vivo, then I think you have, different kinds of plasticity that may be occurring, but maybe at the epigenetic or chromatin accessibility level or DNA methylation level that can dictate kind of the ability of the stimulus to subsequently drive that activation. And so you're really, biasing towards plasticity and specific outputs or, afferents coming in that can kind of dictate how the animal will encode information next time. And I think the really important thing about drugs is that, talking about the signal, okay, let's say, we're talking about a reward. So if there's an unexpected reward, you have an increase in dopamine. And so as the animal learns that a cue predicts the reward, you don't have an increase to that reward anymore. The difference with drugs is they pharmacologically increase dopamine. So every time you take them, your brain is saying, whoops, unexpected reward, better than expected. And so it's increasing your subsequent learning, because every time you take the drug, it's saying that was better than expected. That was better than expected. And so it's increasing the kind of strength of that cue reward association artificially over and over and over again. And the number of kind of associations is really important for how strong that is. And so you kind of have two things at play. One, that it happens over and over and over. And two, that no matter how many times you've taken the drug, dopamine is still going up, even if it's less. In addiction, you can become tolerant, but you're still getting increases in dopamine that are above and beyond that baseline level. And so I think that what you're thinking about isn't just that drugs, you take them and it feels good. It's that you take them and it's artificially driving this really specific learning process that's causing plasticity later. And so, people can stop taking drug, but this plasticity has already happened. And so how do you reverse that? But the plasticity process is a critical process for normal learning. And so I think it's kind of it's one of those problems that it's very difficult to deal with for that specific reason.
So no matter how many papers you publish, whenever you get another one, it feels really good.
Yeah, exactly.
Same thing. So it's
always unexpected.
It's always unexpected.
It's always unexpected. That's the key.
Exactly.
Wow.
Yeah, exactly. Yeah, and so the rest of this paper, I think those are the kind of key things, but the rest of the paper is also looking at kind of, more complex features of kind of these dopamine neuron firing properties. And so, there's a really simple story. It goes up to the reward. It goes up to the cue once the reward is learned. But there's also more complex information being temporally encoded in these populations. And so, in the last figures, what they're looking at is looking at how certain dopamine cells are firing in between that cue and that reward. And so, one of the really interesting things in the learning theory field is looking at probabilities and how they help animals allocate attention. And so things that are really high probability that what's going to happen, you don't necessarily need to pay close attention because you say, okay, that's happening. Things that are really low probability that probably won't happen, you don't need to pay close attention because you're like, that's irrelevant information. But things in the middle where there's a little bit of uncertainty and it has been rewarded in the past, but you're not 100% certain, that's where you need to kind of allocate attention to these kinds of stimuli and say, okay, there's something that's potentially important here. What's going to happen? And so if you look at these kinds of patterns of activation in these kind of median probability areas, dopamine neurons, they go up to that initial cue, but they also have a certain population of these, I shouldn't say all of them, but a certain number of them also have sustained activation that kind of ramps up to the time at which this reward would be predicted and then falls off. And so this kind of dopamine patterns also has been seen in the dopamine release at the terminals, where predicted reward proximity actually leads to ramping up of dopamine release that goes slowly up as animals approach a predicted reward. And so dopamine is really important because it encodes this information in this really kind of simple on-off pattern. But there's also temporal dynamics in between these patterns that can kind of dictate, how animals are attending to different stimuli or allocating kind of effort. And so there's a lot of data from, there's the reward prediction side of the dopamine neurons that people are like dopamine neurons encode this kind of reward prediction error. But there's also quite a bit of data on the other side saying that dopamine neurons encode kind of salience and novelty. So how salient and stimulus is. And what salience and novelty do is they alter our attention to different stimuli in the environment. And so, these kind of dopamine neuron patterns kind of fit with both of these angles. And so these dopamine neurons are really encoding, is something important or not, and how should I kind of attend to these stimuli in the environment? And this is, one example of what's happening in the brain, but there's a lot of data and a lot of different computational labs looking at exactly how that is kind of working in. Jeff Schoenbaum's one I can think of off the top of my head, looking at kind of attention and all of these kind of VTA dopamine neurons, nucleus accumbens, medium spiny neurons, and other populations, and dopamine, and how these kind of fit into these kind of really complex behavioral tasks.
They end up with a discussion of whether reward uncertainty itself has rewarding and reinforcing properties, and they make a analogy with gambling.
Yeah, well, okay, so the best way to get reinforcement is to have schedules of reinforcement that are reinforced consistently but variably.
Yeah.
So the highest rates of behavior in reinforcement tasks are on random interval schedules of reinforcement. So this means that basically you press, or random intervals or random time schedules, but random intervals are better. This means that you basically, let's say, the way that we kind of denote reinforcement is like FR1, that's fixed ratio 1. It means one press is one reward. Mm-hmm. That's stably reinforcing. But if we change it so that on average, every 10 presses is a reward, not just 10, but on average. So some it's 20, some it's 2, some it's 5. That schedule of reinforcement engenders rates of behavior that far exceed any other schedule of reinforcement. And I think part of the reason is that when things are unpredictable, you have to allocate a certain amount of attention to them, and you're not really sure what's going to happen. And so it's most adaptive to actually pay attention and put effort in, as long as it's consistently reinforced. If you remove the reinforcer, then the animals will actually extinguish their behavior. And so it's kind of a close line between consistent reinforcement. It's like inverted U. If you're reinforced at too low of a rate, the animals say totally not worth it. Yeah. If you're reinforced at too high of a rate, the animals are certain, they know what's going on. Yeah. They don't need to allocate a lot of effort, so it's really easy for them. It's that intermediate zone where they have to say, what's going on? How am I supposed to be learning this task? And they allocate quite a bit of motivation and attention, because that's the kind of thing that really drives this behavioral control, and where we should be allocating our effort is in this kind of uncertain environment, but not so uncertain that it's not worth our time. And so this kind of probabilistic decision making is like a really critical component of how we decide where to allocate effort in an environment where we have options.
I guess the solution to gambling is to take away the reward, right?
Yeah, well,
or the uncertainty, yeah.
Or the uncertainty, yeah. Well, I think at some point with the gambling too, and this happens with addiction, so there's a lot of work on these kind of second order schedules of reinforcement where the drugs are reinforced on these kind of really high effort schedules or different spatially distinct where the animals will press on one lever and get drug on the other, so you can dissociate the consumatory event from the appetitive event. What happens over time is that you can actually dissociate the kind of outcome from the stimulus response relationship. And so that when you take, in some situations, like with drugs, when you take away the actual reinforcer, if animals have been working on these kind of really high variable reinforcement schedules, they'll keep pressing for very long periods of time. So I think that in situations like with drugs, by the way, because drugs are not available on an unlimited, whatever you want them kind of basis, or with things like gambling, you kind of adapt these behaviors that cause you to allocate a lot of effort into kind of seeking out and looking for these drugs, and you're reinforced sometimes but not all the time. And so this kind of behavior also leads to these kinds of seeking behaviors that are resistant to extinction. And so, it's kind of a lot of these things, understanding these complex behaviors are a really important component of understanding what the brain does. I'm a behaviorist, so I love the behavioral side. I think a lot of neural people get really excited about being able to record from cells in the brain, but my kind of take is that if you don't understand the behavior, you don't really understand what those cells are doing. Just because something goes up to a reward doesn't mean that cell encodes reward. If something goes up to a reward and a shock, it probably doesn't encode reward. It could encode salience, novelty, surprise. And so like understanding across a whole range of behaviors what cells are responding to is a really critical part of understanding how a cell encodes information in the brain.
And I'll just add, I think the last 5 sentences of this paper are exquisitely written, but the one interesting point is that they say that this, although it's maladaptive in the casino or in the laboratory, this is actually probably super important in natural environments where you want to promote risk taking so you can better understand what the predictive stimuli are. And I'll just go back to my own work, which is that in adolescence, you have an increased risk-taking behavior, and this is potentially associated with differences in dopamine release or dopamine neuron firing. So this system kind of changes across the life and to adapt to different environments.
Right. Yeah. And I think that's another important thing in the dopamine system is it changes over adolescence. And so early on, there's more, actually, and we do a lot of dopamine recordings, there's a lot more dopamine release in adolescents than in adults. And so, also balancing males and females also, and where there's hormonal regulation of dopamine release. And so one of the interesting things about that is that you want to maybe, if you think about evolutionary basis of kind of dopamine modulation for like sex dependent learning, that maybe in certain situations, especially in rodents, when females are able to get pregnant, you want them to actually go out so they can find a mate. But at other times, maybe not as much, because the more you're out in the environment for a prey animal, the more likely you are to actually be killed. And so you want situations where you can modulate how much motor activity or risk taking is happening at specific times to increase the probability that a species is going to be propagated while minimizing the actual kind of adverse effects of being kind of out and about all the time. And so I think it also, these kind of neuromodulatory systems we talked about in the beginning, give a way to kind of modulate the way this information is being kind of transmitted downstream at specific times, whether that be hormonally regulated, stress regulated, satiety state regulated. So one of the things that's really interesting about the dopamine system is that it's differentially modulated when animals are sated versus when they're food restricted. And so, animals are hyperactive, or not necessarily hyperactive, but they're more active when they're food restricted. And so you want to, when the animals need food, to promote them actually taking risks to get food. And so I think it's a kind of a really interesting thing when you're thinking about risk taking and probabilistic decision making, to be able to think about how the system could be modulated in those different cases.
And it's another challenge in the lab where how to model this in rodents, essentially, right? Where they're in a cage with free feeding sometimes, right?
Right. And I think a lot of times in the literature, people don't necessarily, in some fields, the addiction field, animals are usually not food restricted, but then in a lot of reward reinforcement fields, they are. And so how we kind of take a step back with this kind of big picture understanding of the brain and kind of actually go through the literature and say, okay, well, how is this being modulated at different times over development? I mean, the big thing for development is also genetic identity over development. I mean, there's tons of transcriptional changes that happen over development and into adulthood that can dictate different kinds of transcriptional programs. And so, but I think that's
and the response to dopamine changes over development, right? So it's quite a nice complicated system, right?
It's so complicated, but that's why I still have a job. If it was easy, I wouldn't be here. So it's great.
I think that your point about what's happening in the wild is really great, because I was thinking of when animals are seeking food, right? They have to make decisions based on risk and reward,
right?
And they get to learn that over time, right?
Right.
And this is involved clearly, right?
Right.
Yeah, this is fascinating. So cool.
It's really cool. Yeah.
Good stuff. So, This paper is interesting because, unlike the papers in my field, if you go to the references, they have big paragraphs where they talk about things. Like they have one on gambling,
Yeah, no.
Yeah. Well, some of that's changing now. I feel like there's all these journal requirements. All of a sudden, they're like, here, put 10 years of work and you have 900 words.
Yeah, it's crazy.
Yeah.
But now here they let them have like 10 or 12 big paragraphs in the reference section where they kind of expand on things that would have been distracting in the text. I think it's really cool.
Yeah, that is actually really awesome.
So, by the way, this is published in Science, and the authors are Fiorillo, Tobler, and Schultz. They're from University of Fribourg in Switzerland and the University of Cambridge in the UK.
Yep.
It's pretty cool.
Yeah, this is a great, I teach this in all the classes I teach on this because I think it's a relatively simple story that helps understand the complexities of the dopamine system while still being accessible to people outside of the direct dopamine field.
Yeah, do they like it when you teach this to them, the students?
Yeah, it's funny. I'm in a place that there's fewer psychologists. There's a psychology department, but I'm on the medical school side.
Mm-hmm.
And so, I think I look at it as my job to explain to people why understanding how this happens is really important to understanding, what you're looking at when you're patching cells and looking at transcriptional regulation. And so they've been actually very excited about this, but I try to make it relatable to everyday life so that they're engaged. Although sometimes it depends on the class. It's sometimes easier said than done.
All right, before we wrap up, I want to read a couple of emails that we got since the last episode. The first is from Reese, who writes, "Hi, Twinners. I love this new show. I listen to all the other podcasts in the Tweeverse. Love them all. I'm a student at the University of New England here in New South Wales, Australia, where it's sunny and 25°C, making it a lovely spring day, not too hot, not too cold. I'm studying a Bachelor of Science in Biotechnology and Genetics with an aim of getting into research related to bioremediation of climate change, though every time I think I know what I want to do, it changes. So that is definitely provisional." In episode 2 of TWiN, someone said that electrons essentially move instantaneously, which obviously would mean moving at around, though under the speed of light. This is not quite correct, as electrons have mass and are therefore much slower than light in their general movements. The speed of electrical charge propagating through a wire is indeed quite fast, near but noticeably below the speed of light. Electrons themselves are much slower, moving at a speed nearer to a couple of centimeters per second. The way that the charge can move so much faster than the electrons is actually the same sort of process as with waves in water. The wave moves through the water, but what's actually happening is some water molecules are being pushed back and forth with the frequency of the wave, and they pass the force on to the next in the line. Each water molecule moves back and forth around a specific point, drifting over time in one direction or another, but the waves can propagate much faster. A good visual aid for this is imagining pouring some dye in water at a beach. The water will take up the dye, spreading it out over time, but the waves will keep moving through the dye, lifting and crashing it into the sand. The water that impacts the sand with the dye in it will do so a large number of times before the dye dissipates, but the waves that came in that time didn't actually bring new water in any significant quantity. The waves just moved back and forth a bunch of times. Thanks again for a wonderful show. Keep up the good work. I think I said that they moved at the speed of light, so thanks for the correction. Thank you, Reese. Anthony writes, "I don't get no respect. I had the first letter. Should I, like Mendel, simply say mein Zeit word schon kommen?" I don't know. I didn't bother to translate it.
I'm looking.
Look it up. But yes, you did. Anthony did write the first letter. He had said you have a bad link in the episode. So you're right. I do think it'd be great if, as a small regular feature in TWiN, there could be the debunking of neuroscience used as a prop for pseudoscience, like the two examples in the email below. And this is an article he sends called "In Freud on Madison Avenue, Lawrence Samuel reviews the marketing world's brush with motivational research that promised a direct route to the consumer mind, mostly malarkey. The best known is the subliminal suggestion experiment at a Fort Lee, New Jersey drive-in with eat popcorn flashed on a screen. This can be found mentioned in psychology texts today. The only problem is that it most likely never happened. The experiment was claimed by the researcher in the hope of getting contracts from advertising firms.
Oh, yeah.
My impression is that the new neuromarketing is basically the old motivational research with a little recent terminology tossed in. The oft-mentioned reptile brain with its core of fear and greed a rewriting of Freud's superego in id to reach those no longer impressed with psychoanalysis, is for marketers what the philosopher's stone was for an alchemist. Nonsense to peddle to those with more money than thinking ability. Jeepers, with the Twixes flying in so fast now, a Red Queen phenomenon takes effect. I have to listen more and more just to keep from falling behind. Sorry about that, but we're glad you're listening, Anthony. And one more from Caitlin. Dear TWiN Studies, in my previous email, I was referring to how studies on gender in the brain are particularly vulnerable to headline syndrome. They tend to run one of three ways. 1, science proves every stereotype, every last one of them. 2, science knows nothing about gender in the brain. Nothing. It's all a tissue of lies. And 3, surrealism. Some things I have seen include the claim that men think with gray matter and women think with white matter. I know there are differences in utilization, but come on. And some very extreme takes on the differences between the left and right hemispheres of the brain. My personal favorite, though, was the claim that babies do not develop a central nervous system until they are 3 years old. Oh. And until that time, their mother functions as their central nervous system, this being a task that only women can perform. The brain linking ponytails from the movie Avatar come to mind. You don't need. Yes.
What? This is a thing that happens a lot in the sex differences fields. I think that you see it even with things that are close, but I think people really want there to be differences in certain ways. They have lots of preconceived notions. We do some hormonal stuff. And so a lot of times reporters will be like, well, does that mean birth control can cure addiction? And you say no 10 times. And then the title of the story is birth control cures addiction. And you're like, okay, well, I did my best.
A little sanity would be appreciated, or at least some better quality insanity. Well, we will provide better quality insanity.
Yeah, no promises on regular sanity.
Did you look up Mein Zeit?
Yes, my time will come.
Oh, okay. Well, Anthony, you had the first letter. I just didn't remember it because it was a correction. You know, I had a bad link, and whereas Caitlin, who I thought wrote the first letter, wanted better quality insanity, which she explains. All right, that is TWiN number 3. That'll do it for 2019.
Yeah,
hopefully
and the decade. Yeah.
That's the end of the decade, right? And hopefully we'll have 12 twins in 2020.
Absolutely. Yeah.
I'm looking forward to 2020. It's a good year, right?
Even number.
And a new decade.
Yeah.
You can find TWiN at microbe.tv/TWiN. And of course, if you listen on your phone or tablet, which most people do, you're using an app, you can subscribe. It's free. Please do, so we know how many people are out there listening. And you get every episode as we release it. You never miss one. And if you have questions or comments, you can send them in TWiN at microbe.tv. And as I said earlier, if you really like what we do, consider supporting us. You can go over to microbe.tv/contribute. We have ways you could give us like $1 a month. PayPal, Patreon. And you could give more if you'd like, but, you know, $1 would be great. If all of you gave $1, that would help us a lot. Why is that? Why do we need your money? We have expenses. It's not so we can go anywhere, but just to pay our expenses and do some different things. And microbe.tv/contribute. We would love it. If you don't want to, it's fine. It's free. I saw an article this weekend, or an opinion in the New York Times. They're criticizing college textbooks for being too expensive and are not very good. And so I wrote a textbook on virology, which I think is really good. And I know it's expensive, so I don't make my students buy it. I say it's suggested. But I always say, look, I give away all the podcasts. I give away my lectures. I give away videos and blogs. The textbook is one way I make a little bit of money, so I don't think it should be so criticized when professors write textbooks. But we give a lot of stuff away otherwise. And we don't really make that much money on textbooks anyway. Microbe.tv/contribute. Today on TWiN, Ori Lieberman. You can find him on Twitter, Ori Lieberman, one word. Thanks, Ori.
Thanks, Vincent.
And good luck in your transition back to the clinic. Are you now in the lab doing some experiments?
Yeah, I'm trying to resubmit a paper.
Your thesis,
yeah.
Right.
And my thesis, yeah.
So is this your first paper you're going to have, or you've already published some?
No, this will be number 5.
Oh, wow.
Okay.
Yeah.
Congrats.
It's been a fun time. Yeah.
Yeah, well, I'd say.
Yeah.
Yeah.
Erin Calipari is at Twitter, the Erin Calipari. Thanks, Erin.
Yeah, thanks for having me. This is fun.
Thanks for the paper. It was really cool. This is all new for me, and I think it's fascinating.
This is the stuff I love talking about. So I get to learn and rant. That's my favorite thing about this podcast.
Well, I think that for me, understanding what drives human behavior is pretty cool, right?
Yeah, I think that's the thing I like about a lot of the stuff we do is there's a lot of context for why it matters to you as an individual, which I think is the fun part that lets me connect, I think, with people, with my work.
People do like to know why things affect them and why they behave the way they do. The same thing in virology. People want to know why viruses make them sick.
Yeah.
It's always compelling, right? Jason Shepard is at Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent. Yeah, and looking forward to seeing you guys in the flesh next month.
That's right. You're going to be here on January 6th, and you're going to be at that lovely new neuroscience building.
Yeah, it's beautiful.
So it's a new campus they built between the medical center and the Morningside campus because they ran out of space. I'm going to try and get down there and see your talk, Jason, and then
that'll be great.
Then the plan is to do a TWiN up here, right?
Yeah,
that'll be cool. Good.
Yeah, no, never looking forward to it. I haven't actually been to Columbia before, so.
Oh, good. Well, I understand there's some good neuroscientists here.
just a couple, yeah.
So, Jason, it's funny. I had a guest 2 months ago, and he wanted to meet Eric Kandel. He had an appointment, so I brought him down, and I delivered him. And when I was leaving the building, there was a postdoc who I know. When people smoke, they go outside, right? Mm-hmm. So this guy and some people in his lab, they're always outside smoking. So they used to be up here, but they moved down there. Anyway, he said, "Hey, do you know anything about this ARC thing with the virus capsid?"
Nice.
So, because they want to do something with it. I don't know what, but hopefully I'll tell him that you're going and he'll go to your seminar.
Yeah, well, Eric's lab is actually hosting me. So, there's no point in your career where you still don't feel, like seeing that I was going to be hosted by Kandel's lab was, a pretty fun thing to see. So
yeah, it's great.
Yeah.
I can imagine.
That's really cool.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next year.